Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 7, Microbial Genetics. And we have just finished replication, and it was a lot, right? How'd you do on the videos? Did you watch a couple of times? This is one of those big concepts I keep talking about that needs to be talked out. It's going to have to go beyond memorization. You're going to have to understand how things are going. A lot of good videos on this. I'll try to post some like animation videos. But we're moving on from replication because we said DNA has two jobs, remember? DNA has two equally important jobs. One is replication, and that was to pass on to the next generation. So like if we're talking about like grandma's recipe book, right? If we have grandma's recipe book and both you and your sister want a copy, you need to accurately replicate it to be able to pass it along to the next generation. All right, so that's a very important job. The second job is gene expression, because we say grandma's recipe book is valuable because of the recipes it contains. And the recipes is what makes you want her book. It's the recipes, the chocolate chip cookies that you want. So gene expression, it's great if you have a copy of the instructions, but unless you get out the ingredients to make the chocolate chip cookies, you're never going to taste grandma's cookies again. Oh, that's just depressing. Anyway, so you get what I'm saying. Gene expression, that's where you use the instructions and what's in the DNA to actually make a protein, and it's the protein that does the work. It's the proteins that give us our eye color. It's the proteins that make a heart. It's the proteins that do the work. Okay, so let's talk about gene expression. All right, so set of instructions, I said that. The instructions are going to tell you which amino acids to use and what order to use those amino acids to make that particular protein. So just like grandma has a recipe for chocolate chip cookies and she has a recipe for peanut butter cookies or whatever, those are going to have different ingredients and you're going to cook them a different way. So the instructions in the DNA, a different gene is going to give you the instructions to make a different amino acid sequence that gets folded into the correct protein that's going to do the job. Okay, so gene expression, job 2, is made up of two parts, transcription and translation. And remember, we're talking about going from, well, I'll go back to the PowerPoints. The central dogma is what we're talking about, going from DNA to RNA to protein. And again, this is universal. All living cells go from DNA to RNA protein, and this is referred to as the central dogma of that. So going from DNA to RNA is transcription. Remember, C comes before L, translation. So going from RNA to protein. All right, so DNA gets copied into RNA, and then the RNA gets translated into a protein. DNA is transcribed into RNA, RNA is translated into a protein. All right, I do want to point out genotype versus phenotype here. Genotype, if you remember, just reviewing some vocabulary terms that you should know, genotype is what's contained in the DNA. And remember, humans are diploid. We have two copies, one from our mom and one from our dad. So we, in our genotype, we may have the instructions to make brown eyes from our mom and blue eyes from our dad. So that's our genotype. And we have instructions to make both brown eyes and blue eyes. But when those instructions get copied, the instructions to make the brown pigmented proteins, when we actually are mixing the colors and seeing that, it's the pigment from the brown eyes that gets expressed. That's your phenotype. So even though you got instructions on blue eyes from your dad, because the brown gene is dominant, the brown allele of the eye color gene is dominant, then you're going to end up with a phenotype that is brown eyes. So do remember genotype versus phenotype as we're going through that. Okay, so let's look at, this is another picture that I like to look at. So this is the DNA. We've said we reviewed that with the genotype. This is our protein that it's going to make, and that's going to give rise to what phenotype we have. All right, so we had to go from our DNA. Tell me why you know this is DNA. And then tell me why you know this is RNA. What's your clues? Okay, so for DNA, hopefully you said things like it was double-stranded, and we know that DNA is double-stranded. Hopefully you said things like GATCs. Thymine is used in DNA. So these are your visual clues when you get this kind of shorthand thing, this, you know you're looking at a DNA strand because of those things. It's also labeled 5 prime, 3 prime, 5 prime, 3 prime. How did you know this was RNA? Single-stranded, right? And it uses uracil. So again, visual clues. Get used to kind of seeing that so that you can kind of check yourself on some of these problems and see. So DNA would be listed as double-stranded. Sometimes you'll see DNA as single-stranded because it has been unzipped. It has been in preparation for replication. Remember, we unzipped that. So that may be something that you need to know. But that thymine is always going to be present in DNA. Okay, so if we're going to DNA to RNA, we had to transcribe it. And so RNA is only single-stranded, so we didn't have to copy both of these like we did in replication. We only had to copy one. So let me ask you another question. Which of these strands got copied using complementary base pairing to create this RNA? Top or bottom? Okay, so hopefully you said the bottom here, because when we're talking about copying, DNA, either for replication or for transcription purposes, we kind of use that term copy a lot. And if I were going to copy your notes and you wrote down DNA, I would write a D and an N and an A in my notebook. And so it's typical to think of copying as taking the A and putting it down here. But you remember, this is not done by my pen and paper. This is done by an enzyme. And that enzyme uses that process of complementary base pairing, both for replication and for transcription. So which of these was used as the template? The bottom strand. This bottom strand, the enzyme took this bottom strand and used complementary base pairing to create this RNA. So make sure you understand how that works. All right, so that's transcription and then translation. And then again, the RNA is going to give us the instructions on which amino acid to put. And we're going to follow the mRNA starting at the 5' end of the mRNA during translation to put down that to get that. Okay, so let's keep going and get down to the cellular level and figure out how this transcription works. All right, so I've got DNA, and you know it's DNA because it's double-stranded. There's my double helix, right? But this time we want to undergo transcription, not replication, transcription. So I don't need to copy the whole thing. I just need to find the gene that I want to transcribe because I want to make a protein from that gene. So there is an enzyme called RNA polymerase that is going to attach to RNA and scan it. And it's looking for that gene. How does it know it found it? There is a region called the promoter, and that'll be on the next slide. There's a region called the promoter that tells RNA polymerase to start here. And remember, it's an enzyme. It's made after, it's named ACE, right? So we know it's an enzyme. Enzymes are named after what they do. This is RNA polymerase. We're going to polymerize or synthesize, make RNA. So this enzyme, RNA polymerase, is scanning. It's looking for the promoter. When it sees it, it is able to open up. Remember, unzipping, replication, we had helicase. In this case, RNA polymerase is able to open it up at the promoter and then start using complementary base pairing to add in nucleotides. And just like DNA polymerases, RNA polymerases have to add it into this 3' end. So they'll always be adding it to the 3' end. So the RNA, you can say, gets transcribed 5' to 3', and that's because it's always looking at the 3' end. Sometimes books will tell you that RNA polymerase moves along the DNA 3' to 5'. And if you look, if this is the 3' end of my mRNA, and I know it is because that's the only place we can add in nucleotides. If this is my 3' end, that's my 5' end, that's my 3' end, and you can see it's moving down this direction. You can memorize that. That's great. I'm always looking for that 3' end. If I know that the new strand can only be used in that 3' end, then I know that, sorry. Okay, so I am always looking for that 3' end, and then I can figure out where everything's going. Okay, so that's that picture. Let's look at this in another diagram. So we said that RNA polymerase attaches to DNA and it's traveling down until it finds a promoter. So we said replication was origin. For transcription, it's looking for the promoter. And again, this is just a sequence of DNA, G, G, A, A, T in um, uh, a certain sequence that tells RNA polymerase start here. So this promoter is the sequence of DNA, and they denote it in yellow. So the RNA polymerase is looking for that. When it finds it, it will open it up. Now, unlike replication that unzipped it, this just kind of opens it up a little bit. We refer to this as a transcription bubble because after that, before and after, you can see as RNA polymerase moves down, the DNA goes back together. So it's not unzipping, it's just opening it up to form this transcription bubble. Now there's something else that I wanted to talk about. There are certain proteins called sigma factors that can bind to promoter regions and help RNA polymerase find it. So it facilitates attachment and helps the RNA polymerase find it. This is helpful because some proteins you want to make more than others. You need to make a lot of mRNA because you need a lot of that protein you make more than others. And so you're going to use some sort of system to help recognize it. This is, if we go back to the recipe book, these are like your sticky tabs. You know, like chocolate chip cookies, you want to make a lot. So you like fold down the page or you put a little sticky tab on there. That's the purpose of a sigma factor. It's to kind of mark it so that you can easily get back to it. So there are other, proteins you don't need to make as much, so there's not as many sigma factors or any attached to it. So those kind of things. So that's what a sigma factor does. All right, so now we have initiation. RNA polymerase is bound and has opened up this transcription bubble. This is referred to as initiation of transcription. This is what gets it started. So now we need to move, we need to actually make the mRNA, and this is called elongation because that RNA polymerase, once it opens it up, it'll start adding in nucleotides. And remember, this time it doesn't need a primer. It just adds the first one down, and then it always adds to that 3' end. So here's RNA polymerase. It started here, adding a nucleotide, and it's using complementary base pairing to add in more. And you can see that transcription bubble moves down the DNA as it's going through that. All right, so only copying one, and we refer to that as the sense strand. I don't think I said that earlier. The template or the sense strand is the one that is getting used as a template to create that mRNA. Just like replication, the nucleotides that are added, these we're making RNA, so they're ribonucleotides versus deoxyribonucleotides, and even more specifically, they are triphosphate ribonucleotides. Why is that? Yes, because of the energy. You can add this in. It's a building an anabolic reaction. You're going to cleave those two phosphates off to build this. All right, so this is considered elongation, and that will continue, but it doesn't need to continue forever like replication. So we need a stop. And so the stop region or the signal is called a terminator. So RNA transcription starts at the promoter and proceeds until it gets to a terminator region. And again, it's just a sequence of different DNA. There's actually a couple different terminators, just like there's a couple different words for the word stop, terminate, halt. There's a couple of different ways that we can terminate, so sequences that happen. So two ones that happen in prokaryotes is a self-termination, and it comes up on a sequence where there's lots of repeated G's and C's. And when that happens and the RNA starts putting down a whole bunch of G's and C's, well, RNA is single-stranded. Those G's and C's kind of want to bind to each other. They want a hydrogen bond. So they end up forming this little loop. The mRNA, you see the red mRNA, forms this little loop. And then that's enough to pop that RNA polymerase off. So it disassociates from that. Another way that we can get termination is by adding a protein helper. This one is called Rho, R-H-O, and Rho binds to a sequence on that, and it kind of wedges itself in between there, and it also causes the RNA polymerase to come off. So the R binds to a certain sequence in the terminator region, and then that when the RNA polymerase gets there, it causes it to come off. Okay, so those are both ways of RNA. Now remember, this is a prokaryotic cell, no nucleus, and in fact on the test, if you identify that as a nucleus, I put like in the nucleoid region is a nucleus. I wrote it in capital letters. There are no nuclei in prokaryotic cells, which means transcription is occurring in the cytoplasm, replication is occurring in the cytoplasm, so is translation. So anyway, well, that's coming up. But so everything's occurring in the cytoplasm. All these enzymes are in the cytoplasm. Once the RNA polymerase binds to the promoter and the transcription bubble moves down and opens up the next section, you see that it went back here and the promoter is intact again. It went back together, which means another enzyme, another RNA polymerase can bind to that promoter. So during transcription, you actually see multiple RNA polymerases going through and multiple mRNA transcripts being done. So that happens. This actually happens in eukaryotic cells as well, because it's all happening in the nucleus. All right, so here's your practice. I am giving you a double-stranded here. You know it's DNA, right, from those clues. I'm going to ask you to transcribe the blue strand of DNA into mRNA. So the blue strand is the sense strand. So go ahead and do that. Did you get your pen and paper out? How'd you do? Okay, so hopefully you were able to use, if I tell you that the blue strand is the sense strand, you were able to use that and use complementary base pairing to put down. You know you're making RNA because I've asked you to transcribe it, transcription. So you used uracil. So you made a single-stranded copy and you labeled it. If this is the 3' end, you know this is the 5' end. So you started here and went this direction, and now you have an mRNA, and this is the 5' end. You should be able to do this. Okay, we're going to stop here and pick up the next video, but if you'll notice, these are usually written in groups of 3. Now, this is nucleic acids. There's no space here. Why do we write this in groups of 3? That's kind of a teaser for the next video. So, all right, I'll see you at the next one. Thanks.